Hello everyone, welcome to this channel. This is Rahul Roy. Just like a country is identified by its flag, do you know our bodies RBCs are also identified by its own flag? So in this video, we are gonna talk about ABO blood grouping. For this, I have prepared a presentation to understand the science behind it. Here in the picture you can see the red blood cells, and some of the red blood cells are having some protruded structures. These are nothing but antigens. Genetics of ABO blood grouping. The ABO gene is autosomal. That is, the gene is not on either sex chromosome. ABO blood groups are regulated by the gene I. The gene is located on chromosome number 9. ABO blood grouping is based on the presence or absence of two surface antigens. Antigens are nothing but chemicals that can induce immune response on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies, and antibodies are nothing but proteins produced in response to antigens. The distribution of antigen and antibodies in the four groups of blood A, B, AB and O are given in the following table. So let us look at this table. Suppose an individual having blood group A, that is antigen on its RBC will be A, and anti-B antibody in its plasma. As anti-B antibody is present in his or her plasma, so a person with blood type A and O only can donate to that individual. Now, if a person with blood type B having anti-A antibody in his or her plasma, so if he or she donate blood to that individual whom we considered firstly, there will be a process called agglutination, that is antigen antibody reaction, which will bring destruction of RBCs. By applying this concept, you can identify the donors group of other blood types. Similarly, we can see here, if blood group is O, that is, it does not have any antigens A or B, but it will have antibodies like A and B in its plasma, and the donor group will be only O type. During blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of the recipient before any blood transfusion, to avoid severe problems of clumping. Clumping is nothing but destruction of RBC. From the above table, it is evident that group O blood can be donated to persons with any other blood, and hence O blood group individuals are called universal donors. Persons with AB group can accept blood from persons with AB as well as other groups of bloods. Therefore, such persons are called universal recipients. Generally, A and B blood groups are dominant over O blood group. Each person has two copies of genes coding for their ABO blood group, one maternal and one paternal in origin. The plasma membrane of red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. This is nothing but the antigen. Here you can see the picture. The antigens are present on the plasma membrane of RBC, and there is an interaction is showing antigen antibody interaction. The gene I has three alleles, IA, IB and I. The alleles IA and IB produce a slightly different form of the sugar, while allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over I. In other words, when IA and I are present, only IA expresses, because I does not produce any sugar, and when IB and small I are present, IB expresses. But when IB and IA are present together, they both express their own types of sugars. This is because of codominance. Actually, IA and IB both are dominant, so both express their own types of sugars. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood types. So from here we can also find the number of phenotypes, possible phenotypes. So based on the above understandings, we can make a table showing the genetic basis of blood groups in human population. Suppose allele from parent 1 is IA and allele from parent 2 is IA. So the genotype of the offspring will be IA, and the blood type of offspring will be A type. Similarly, when IA from parent 1 and IB from parent 2, the genotype of offspring will be IA, IB. As a result, the blood type of offspring will be AB type. Similarly, again we can see, if parent 1 having IA allele and parent 2 I, the genotype of offspring will be IAI, and the blood type of the offspring will be A type. Similarly, IB and small I from parent 1 and parent 2 respectively, the genotype of the offspring will be IBI, and the blood type of offspring will be B. When parent 1 possesses small I allele and parent 2 also possesses small I allele, genotype of offspring will be II, and the blood type of offspring will be O type. That is, it does not produce any antigen, but it will have anti-A and anti-B. So 6 genotypes are IA, IB, IB, IA, IB, IAI, IBI and II. 4 phenotypes are A, B, AB and O. Phenotypes are physical appearance, and genotypes here means genetic appearance. I hope you get the concept behind ABO blood grouping. Thank you for watching the presentation. If you like the presentation, give a thumbs up to it and give your valuable comments. And lastly, if you find it informative, please share and subscribe the channel as well. Thank you.